Hello, everybody. Let's give 1 minute before so everyone can join and we can start. Okay, good morning, good afternoon, good evening, everyone joining us from different parts of the world. Welcome to this Marine Biodiversity Networking Friday session organized by Air Center and MBON. And my name is Pericles Klitou, and I'm a marine scientist at the Marine Environmental Research Lab based in Cyprus, that is in Eastern Mediterranean. I work on marine biodiversity, non-indigenous species, fisheries management, sustainable aquaculture, and stakeholder engagement. Much of my work has been focusing on biological invasions in the Mediterranean Sea. So today's topic is especially relevant to both my research and the wider marine biodiversity community. Today, we are discussing marine biosecurity and the need to strengthen collaboration, the communication, and standardization across marine invasion networks. So biological invasions by non-native species are not only an ecological issue, they can affect fisheries, they can affect aquaculture, food security, local economies, human well-being, and coastal communities. They are also connected to wider global processes, including shipping, trade, climate change, and the changing of species distributions. At the same time, marine biosecurity remains a difficult challenge. In many regions, we're still trying to understand not only which species are arriving, where they're spreading, but also we want to understand their impacts, how they interact with the native communities, and what kinds of management responses are realistic. So this is where today's presentation is especially relevant. We hear about the BRIDGE program, which comes from Bridging Networks to Advance Collaboration, Communication, on Marine Biosecurity. BRIDGE stands for Bioinvasion Research Integration across dynamic global ecosystems. The program aims to connect regional and global expertise, support standardized approaches for detecting non-native marine species, improve data sharing, and build the collaborative capacity needed to advance marine invasion science and biosecurity policy. For me, one of the most interesting aspects of this topic is how we can better link observation systems with decision making. Detection and standardization are essential, and we need to move towards understanding impacts, ecosystem change, and threshold for actions and practical response options. To guide us through the topic, we're very pleased to welcome Dr. Amy Freestone. Dr. Freestone is a senior scientist at Smithsonian Environmental Research Center and the managing director of the Marine Invasions Research Laboratory. Her work explores the patterns, processes, and consequences of invasions in marine ecosystems from local to global scales. She joined the Smithsonian in 2024 after 15 years of faculty at Temple University, where she was also the founding director of the Temple Ambler Field Station. She completed her PhD in ecology at the University of California, Davis, and was previously a postdoctoral fellow at ERC. It's exciting to have her talk today, and please feel free to use the Q&A box while she talks about non-native species. Amy, thank you for joining us today. The floor is yours.
Great. Thank you so much, Pericles, for the lovely introduction. I feel like I don't even need to give a talk now. I feel like you summarized it all. So thank you so much for that. And thanks everybody online for joining. It's really a pleasure to be here and talk about this. I'm the principal investigator of the Bridge project. I'll tell you more about that. But I want to highlight from the beginning that this is very much a collaborative effort, including collaborators across MBON. So it's really great to be here. So let me just take a moment and share my slides. So hang tight. All right. You guys see okay? Correctly? All right, great. So yeah, I am the managing director of the Marine Invasions Research Laboratory. So we are at the Smithsonian Environmental Research Center in Edgewater, Maryland. We're a staff of over 40 scientists. And we are based in Maryland, but we also have a satellite lab out in Tiburon, California. So we have staff on both sides of the continent. And a lot of our staff have been with us for decades. And so we have a very deep expertise in the field of marine invasions, which allows us to really engage and do cutting edge science in this field. A little background, but I feel like Pericles already hit the high points, but I'll reiterate. Across all biomes, so we're here talking about the oceans today, but across all biomes, invasions are a primary driver of global change. Many of us know this. They can have substantial ecological impacts. They can crowd out or consume native species, modify patterns of biodiversity, as well as ecosystem function. They've contributed to 60% of global ecosystems, and they affect critical infrastructure that we depend on in our society. Pericles already mentioned some of these fisheries, local economies. They support, they can impact critical infrastructure that support our municipal drinking water, delivery systems, hydroelectric power, agriculture, marine operations. We can keep going. Many of us know the impacts of invasions are vast. They're also expensive. So invasive species broadly, their costs have been increasing, as we see here, 4 times every decade is what we've seen. And currently, costs exceed over $400 billion every year across the global economy. And this includes losses to people's quality of life. So we see here invasive species have a negative impact on good quality of life in 85% of cases. So this affects all of us in very different ways. For marine invasions in particular, and that's what I'm going to focus on today, our primary vector for marine invasions are commercial ships. So commercial shipping, of course, is critical to global trade. But with that exchange of goods that we need across our global economy, we also get a lot of hitchhikers joining those ships. So there's 2 primary ways that species are transported by ships. First is ballast water. Ballast water has historically been a primary driver of marine invasions. This is water that ships take on in one port to stabilize themselves in transit. They discharge that water in another port, depending on their load. And of course, that ballast water is not just water. Lots of things can be transported in that water, including live organisms. And so you might say, well, how much ballast water is actually moved around the globe? The answer is a lot. In just one bay in the US, I'm going to highlight just here San Francisco Bay, one of our a very busy hub in the US. Every year in San Francisco Bay, we can have over 1 billion gallons of ballast water discharged. That is over 1,700 Olympic-sized swimming pools every year. And of course, that's a lot of water. And there could be, busier ports are going to get more, less busy ports are going to get less. But any way you slice it, that is a large amount of ballast water that's being moved around globally. The other way that species can be transported by ships is on the hull, the bottom of the ship. So we call this hull fouling. So ships, and these commercial ships can be enormous. A single tanker can have over 7,000 square meters of surface area below the waterline. That's larger than a football field. This is huge. And even when you think about the size of some of these marine organisms, and these tiny species, one, they can grow quite happily on hard substrate, like the bottom of a ship, many of them can. And they can have life stages like the size of a pencil point. And so a football field, or even a fraction of a football field, can be an enormous amount of habitat per ship. Times that in the US, tens of thousands of ships that are coming into our port every year. And of course, that's replicated across the globe. In the US and in many countries, we have some biosecurity policies that help us reduce our risk of invasion. And in many countries, including the US, ballast water is required to be treated to comply with International Ballast Water Management Convention, with guidelines that have been established by the IMO, the International Maritime Organization. And approved and certified ballast water management technologies are now widely adopted across the global fleet. And advancements are now being made to consider guidelines to reduce the risk of invasion by hull fouling. But a critical question remains, are these policies working? Are they enough? So on behalf of the US, our lab hosts the National Ballast Information Clearinghouse. This is a collaboration between the Smithsonian Institution, my institution, as well as the US Coast Guard. And so all ships in the US, if you come into a US port, that ship is required to file a ballast water management report to the clearinghouse. So we receive, process, and analyze over 100,000 reports every year. We synthesize those data and we make them available to our Coast Guard, to local and state governments, as well as to the public. And we study shipping patterns in key regions in the US as well as abroad. So here's shipping patterns for the Panama Canal shown here. We also study ballast water itself and what occurs in ballast water to ask the critical question of, are the treatment systems that ships are required to have, are they even working? So our teams will board these ships, take samples directly from the ballast water, and then sample them live for live organisms to understand whether they are meeting those discharge standards. So our discharge standards require a relatively low level of organisms per square meter or cubic meter or metric ton of water. But even a few live organisms in a metric ton times the millions of metric tons that are being moved around is still a lot of live organisms moving around. So it does not eliminate risk, although it reduces risk. So invasions can still occur. So we also look for invasions, right? We also survey, which is an important critical piece in understanding invasion dynamics when we think about detecting them. So we have surveyed over 30 bays in the US and more internationally. We also have a series of sentinel sites shown here in the lower right, those gold stars, where we go back repeatedly to the same bay to understand trends through time. You'll notice here we are the Marine Invasions Lab, but we also study some freshwater systems, including the Great Lakes. So we kind of span the salinity gradient, as it were. So we use our survey data, our standardized survey data, and we'll touch on that a little bit more later. We leverage those standardized data as well as other data that we glean from the literature and other efforts to track the status and trends for invasions for the US. And there's over 500 non-native marine species of invertebrates and algae in the US right now. So we synthesize those data and then we make them publicly available through an online portal that's widely used by researchers, policymakers, managers, as well as the public. And we also host separate portals for key international regions like the Galapagos and Panama. But this is not a US issue, right? This is an international issue. And in fact, in 2022, the Convention on Biological Diversity set the very ambitious but critical goal of reducing the impacts of invasion and reducing the rate of new invasions by 50% by 2030. So this has rallied the international community, which is a fantastic thing. So this was set back in 2022. It's now 2026. We're supposed to meet this goal by 2030. We are halfway in with a whole lot more work to do. So clock is ticking, but we have our work cut out for us. So there are challenges to address this goal. And this brings us to knowledge gaps, which Pericles mentioned earlier, the things that we don't know that are holding us back from really making advances in this field. So for marine invasions in particular, there are a lot of knowledge gaps. Bioinvasions and biodiversity, marine biodiversity broadly, as we know, is underreported. We don't have enough understanding in key regions around the world. And I put up this figure because I think it highlights a couple things. One, what we know as well as what we don't know when it comes to marine invasion. So this is the number of aquatic non-native species that was put out by a really lovely paper in 2020 by Sarah Bailey and colleagues. And you can see here the heat map showing numbers of non-natives in different regions of the world. You can see really high levels in the temperate zone. So in the US, in the Mediterranean, Pericles, where you are, you can see that those latitudinal gradients of non-native species, again, with temperate zone really being a hotspot. You can see fewer invasions happening at high latitudes. We think that is likely a real signal, although also underreported and changing, as a lot of our ecosystems are changing with different dynamics, including global change and changes in trade dynamics. The tropics are tricky here because we see the tropics, and in some regions, largely in the global south, we have that white zone, data not available, right? We do not have good data in large chunks of the world. And so this really reduces our ability to understand large-scale patterns of invasion. So these data gaps are critical for us to fill if we're going to understand these patterns. But we do start to see some of these patterns emerging about where those hotspots are and how those can change across large global gradients. But there's still a lot more work to be done to understand these patterns. But I wanted to just step out of the marine realm for a moment and say this is not just a marine issue, our data gaps in understanding invasions. It's actually an invasions issue. So here we have a study for global terrestrial invasions by Chong et al. In 2021. This is what they show here is the number of studies. Each of these pie graphs are showing the number of studies on different terrestrial taxa. And what we can see here, again, is a very similar pattern that we know a lot about temperate systems. We know a whole lot less about tropical systems and not as much about our polar systems as well. So again, those same patterns that we see about those knowledge gaps are emerging in the terrestrial literature as well. And I'll give one other example of this, for plant invasions, again, it's easy to think that we know more about plants and terrestrial and ecosystems than we do about marine systems, which is probably true to some extent, but there's knowledge gaps there too. Again, these pie charts showing the number of studies for plant invasions. Again, you see these big swaths of the globe, including the tropics and chunks of the global south that are just simply undersampled, understudied. And we can see how that plays out with the figure on the right. Again, most of the study is occurring in the temperate zone and in subtropics, Mediterranean, a little bit in what they call cold weather, those high latitude or high elevation sites, and again, a small sliver in the tropics, just highlighting how much we don't know. But of course, understanding invasions in any biome is complicated. And so there have been lots of conceptual frameworks that have been offered over the years. This is one. I could show you a slide with a dozen others. This one I like. Each of these bubbles, you don't need to read it. Don't worry, don't strain your eyes. But each of those bubbles are a hypothesis for invasion biology. And a lot of those hypotheses have been very critical to shaping our understanding of invasion dynamics. And so this study has tried to link them conceptually and highlight areas of related hypotheses, which is helpful. But as we learn more about invasions, particularly across large global scales, we realize that that invasion dynamics play out differently depending on where you are in the globe. And some of those patterns are actually predictable. So we're beginning to increase our understanding of how those different dynamics play out depending on where in the world you are. And that understanding has not yet reached our conceptual frameworks. So we need to rethink some of these frameworks that structure our field to integrate the current knowledge in how invasions play out across the globe. So how do we tackle these grand challenges that's kind of defined by the Convention on Biological Diversity? We need a global approach to marine invasions, right? We need to improve our conceptual frameworks to integrate our understanding about global dynamics of invasions. We need to be conducting coordinated experiments that allow us to understand ecological dynamics at scale, which is not a minor thing. We'll talk more about that in a moment. We need to increase the breadth of our detection and alert systems, knowing where the invasions are occurring. And again, as Pericles mentioned in the beginning, making sure we have a pathway to actionable science on that, getting those information to managers and policymakers. And again, that brings us to that fourth piece of using robust science to inform sound policy and sound management practices in the field of biosecurity. So this is why we established BRIDGE. So BRIDGE is, as Pericles mentioned, the Bioinvasion Research Integration across dynamic global ecosystems. And we are just launching BRIDGE now. This was funded by the United States National Science Foundation last year. and it is a network of networks. And it's meant to accelerate science in the field of marine invasions. So some of our broad goals are to create a platform for collaboration and communication to accelerate the science, to standardize methods of data collection, management, and sharing, to coordinate experiments, really key to improving our understanding, and again, to mobilize that science into action. And remember, the scope here, while we draw on our understanding from freshwater systems and from terrestrial systems, the scope of BRIDGE is really marine. We're looking at marine and estuarine ecosystems here primarily. So we are a network of networks. So we are leveraging the skills, the training to address some of the needs in this area across global networks that are either focused on marine bioinvasions or marine science broadly, like MBON. And every network has their own expertise that they can bring to the table. And a lot of networks have needs that we can help address through BRIDGE. So here's an image just of some of our initial collaborating networks, but we're just getting this initiative going, and we are happy to have other collaborators. So we anticipate and hope and expect and want this initiative to grow with time. So if you're a part of another network that you think is a good match for us, please let us know, because we are interested in continuing to expand. And whether you're a member of a network, and you'll see NBON here, so many thanks to our NBON partners here. But what we're hoping is, even if you are here, a member of the public, whether you're affiliated with a partner network or not, the goal of BRIDGE is really meant to provide resources and build capacity across the global community to advance the science and action related to biosecurity and bioinvasions. So we lean on these partners, but we're not exclusive to those partners in terms of the reach of what we're trying to do. We are, the funding that we have, our initial funding, is for networking. So we can fund meetings, we can fund trainings. I'll tell you a little bit more about that. But what we're trying to do is really focused around 3 research themes initially. We hope BRIDGE will continue on into the future, but at least for the first 3 years of our project, we had 3 research themes that we think will start to springboard the field in positive ways. So the first is understanding invasion patterns, as I mentioned, resolving invasion extent at scale, understanding where invasions are occurring, and improving our knowledge, filling some of those data gaps so we can get a better handle on where those invasions are occurring. This is not in and of itself, this is not a minor piece of what BRIDGE needs to do. The second themes are related. So the extent is where they're occurring, but of course, they occur there because of kind of 2 suites of mechanisms, right? One, how they got there, the propagule supply. And so theme 2 is understanding propagule supply through synthesized vector analysis across scales. So again, the movement of species around the globe. And theme 3 is understanding the ecological dynamics that determine whether those propagules are going to be successful or they're going to fail. And so that includes biotic interactions, that includes environmental variables, whether that's invasion resistance or susceptibility to invasion, understanding those ecological dynamics that are going to dictate the pathway of that invasion. And of course, all these research themes through what we're doing are meant to integrate again with management and policy throughout. So how we're structured, I just want to give you a little bit of an overview of how the program is structured. We have working groups that are tackling each of these issues. Across the first 3 years of BRIDGE, we will be launching 6 working groups. We have 4 working groups that are active now. Again, we're just getting going, but we have 4 that are active now. 3 we consider operational working groups that we expect to continue on through the lifespan of BRIDGE. And then 3 are thematic, hitting at those research themes that I mentioned. So we have a communications working group, and I'll tell you a bit more about each of these. Communications, training, data management and sharing. Of course, those are all 3 pillars that are critical. And then the theme for year 1 is detection, again, getting to extend understanding where invasions are occurring. And in upcoming years, we're going to get to hitting at those other research themes, running coordinated experiments to understand some of those ecological interactions that are structuring invasion dynamics, and then understanding propagule supply through a vector analysis. Just a little bit more about our leadership structure. We have a steering committee. So this is composed of the senior personnel who are originally part of the BRIDGE grant. So all these PIs, including myself, are from the Smithsonian. But also on the steering committee are representatives from each of our partner networks. And those, again, are really important, critical partners to understand, to making sure that our activities are well informed, and we're bringing voices from around the globe to help structure our thoughts and our activities as we move forward. So our steering committee supports the goals of Bridge through advising us on direction and approach and facilitating exchange of expertise within their network and across networks within Bridge, and support communication with the memberships of their partner networks. We also will be developing an advisory board with members of academia, management, and industry to make sure that we are engaging across sectors when we think about the strategy of Bridge and the long-term sustainability beyond the lifespan of our initial funding. Our timeline, as I've said, we hope Bridge will continue on in the future, but we have funding for about a bit over 3 years to get us going. So this is an arrow in the sense that we hope it will continue on. We always have long-term sustainability in mind when we're structuring our activities. But I want to take you through some of the events that we're going to be having over the next 3 years. Every year we will have an all-hands meeting to facilitate communication with the broader global Bridge community. These are open to anybody who is of interest. And this is an important opportunity to engage with that community and to get feedback. We just had our all-hands meeting for this year, and on April 29th, we'll have it in April, spring of every year moving forward. So if you missed our April 29th one, please come and see us again in the future, and we'd be happy to share notes from that meeting if you're interested. Every year, we will also be hosting a workshop that will focus on each of those research themes that I mentioned. So these workshops are primarily for our working group members, some early career scientists. Again, we have an eye for training, and technical experts that can help us dig into different topics. So the focus of these workshops are to fill critical gaps in the field and engage on emerging topics. So year one, again, our research theme for this year is detection, thinking about invasion extent at scale. And that workshop will be held this September at the Smithsonian Environmental Research Center in Maryland. We will also, after each workshop, we'll be having an in-person training. And this is primarily for early career scientists as well as mid-career scientists who may be in a good position to start to engage in a specific topic. So again, this year we're focusing on detection. And so we have folks coming to our training in September, again, from early career all the way up through more mid-career senior people who really have a program where they can advance detection efforts. So this is about workforce development and focusing on technical skills. And again, year one is focusing on detection. I'm going to tell you a little bit more about exactly what we're going to be training on. But of course, to have in-person trainings, we can't bring everybody across the world to these trainings, although in-person we know is so valuable. But we supplement this, of course, with virtual trainings to hit a broader group of people. So these are open to the public. These are intended to build capacity on specific skill sets. We have these here slotted in for every fall to kind of have virtual modules that can supplement those in-person trainings. But this year, we anticipate rolling out some of these virtual modules as early as this summer, July, August in the Northern Hemisphere. So keep an eye out for these virtual modules as we roll them out this year and in the future. We will also be having a fellows exchange. So these are research exchanges specifically for early career scientists. This is intended, again, to promote workforce development, focusing on technical skills, giving young scientists or early career scientists an opportunity to study somewhere else, whether those are scientists in the US who want to go abroad, or scientists in who are in another country and want to come to the US. These research exchanges can be a really, really critical platform to expand early career scientists' understanding of new technologies, new approaches, and for them to network themselves and hopefully to create some valuable contacts for their careers and hopefully advance them in the field of marine bioinvasions. So our communications working group, I'll tell you a little bit about each of our working groups now. Communications is a critical piece for what we're trying to do, obviously. We are trying to share our goals with the global community, which is why I'm with you today. And we are already reaching groups all over the world, which is really exciting. And of course, we have a lot more work to do here. But the map that I show is just some responses. So these are the numbers that you see in those circles are the numbers of people who completed a survey that we put out with our all hands meeting this year about where do you work? Where is your study of invasions occurring? And so you can see that we're hitting on lots of different major bodies of water. That 7 in the corner is just other. It's not extraterrestrial invasions, but we have really hitting a lot of different marine and estuarine ecosystems all over the world. We are very aware that there's care that needs to be put into how we communicate. So for all of our communication platforms, we are thinking about language translation. Our all hands was offered through Zoom with multiple many opportunities for closed captioning in many different languages, and we'll be providing materials in different languages as well to try to again engage the global community. We are developing a website. I wish I could give you that link now. It is in development. We're close, but the website is coming soon. So please keep an eye out for that. We'll also be, once the website's going, we'll also be launching our social media platforms, so you can connect with us there. When we will be doing a newsletter as well. We're also rolling out outreach at different conferences and taking advantage of opportunities like these networking Fridays to try to get the word out. We have an email account, bridge@si.edu. So I'll pitch that to you at the end. If you have any interest at any time in getting involved, you want more information, you want to know when our website gets live, you want to be on our mailing list, please email us at bridge@si.edu, and we'd be happy to stay connected with you. Our detection working group is another critical piece for what we're working on this year. Again, the intent here is to resolve invasion, or I say move the needle in understanding invasion extent at scale. And so we're basically taking 2 approaches in this working group. One, we're starting an effort to synthesize detection approaches to create a roadmap for users. There are lots of ways you can detect non-native species. So we want to organize some of that information for people who are engaged in this field, try to show people what your options are for different habitats, what resources you need to be able to do different types of approaches, the strengths and limitations of different approaches that you may use to detect non-native species. Highlight whether they're ready to be whether these approaches are ready to be adopted at scale. Some are ready to go, and I'll mention one in a moment. Some are not, but are really exciting. So we want to see where those different approaches are, and of course, when they're ready to be used at scale and deployed at scale, then we need to be very careful that we're all doing the same thing, and that gets to standardization. And I'll talk more about that in a moment. And of course, to keep an eye on emerging technologies. We all know eDNA is really hot right now. There's other emerging technologies that I think will really help advance the field of invasions and detection efforts. So we're keeping an eye on that, recognizing that not all of them are ready to go to scale, but we should keep an eye on them as we move forward in this field. But in terms of providing standardized detection protocols, this is another key piece of what the detection working group is trying to do. So again, I think this may go without saying, but just to make sure that we're all on the same page, the value of having standardized protocols is that we can address gaps in data coverage, but have that done in a way that we can directly compare those data. Otherwise, that's very, very difficult to do. Of course, any field protocols that we have, data collection protocols we have to be paired with, have to be integrated with data management and sharing protocols, right? So you collect good data, and then you get that data out when we share knowledge across the global community. And all of this is working towards a coordinated detection and alert system. So year 1, this year, we are focusing on standardized protocols for fouling panels. Fouling panels, for those of you who use them, this may be repetitive to you. Those of you who don't, I'll give you a quick primer on fouling panels. There are lots of ways, as I said, to detect non-native species. Fouling panels are one that are already in widespread use, and they're very easy to use, which is why we're starting here in terms of our standardized protocols. But we're not ending here. This is just the first step in rolling out a series of standardized protocols for different detection efforts. But fouling panels is an easy place and a good place to start. So these are PVC plates that you can put out in your coastal environment. These plates are really elegant in that they allow us to standardize the substrate, they standardize the area, we can standardize when we deploy them, when we retrieve them. They're inexpensive and they're easy to use, and then you can put them in the ocean, you can have a community that develops And then you can remove them and bring them back to the lab and study them. And they're very easy to put in ports and marinas, places where we know there is a lot of non-native species that happen to also like artificial habitat, which the plastic panels are good for. So we're starting with these fouling panels. Our intent with these protocols and this intent will be true for all the protocols that we develop through Bridge, but to keep the bar for entry low. We recognize that labs, people have very different levels of resources, have different levels of expertise, and we want everyone to be engaged to the extent that they can. So we want to make sure that the protocols are accessible, they're easy to use, they're clear, and that bar for entry is low. For plates, for these fouling panels, we understand that people have different coastal environments where they might want to deploy these. So we're going to have different modules where you can deploy fouling panels on a dock or on an benthic environment, depending on what you have locally. We'll also be deploying protocols or modules that will allow you to assess that panel community morphologically, as well as using genetic approaches. Morphological approaches are going to be certainly the cheaper option, but genetics, as we know, it's great to have sort of that integrative approach to detection. But we'll have both options available, again, standardized protocols with accompanying training modules. And then with this coordinated detection that we're trying to ramp up now, this also gives us an opportunity to do studies on top of that. And again, we're trying to accelerate that research piece, not just understanding sort of where invasions are occurring, but really facilitate other types of questions. And so we're already having interest across the global community for people who are like, oh, I could study this and I could study this. And I think, fantastic. Tell us what your ideas are. If there's something that we can do to layer onto these protocols and other people want to play, then fantastic. Then we can facilitate those kinds of coordinated studies. And one of those coordinated studies that we've been active a lot in our lab is about predation and caging. So these panels are great because, well, one of the reasons that they're great is that you can easily cage them, and these cages can keep out predators. So you can have some panels that are open to predation and some panels that are closed to predation. And we know that predation in some regions, particularly in some regions of the tropic, can be really, really strong. And that can actually impact the invasions that you see. I include a picture here of what a plate looks like. The one on the right had a cage around it, and the one on the left did not. So you can see just how strong that predation signal is and how much biomass is being consumed by predators. There's all kinds of cool ecological questions that surround predation and how that changes across global dynamics or global gradients. But it's also important for detection. In our studies, we found that some species we didn't even know occurred until we put a cage around the panel and found them because they were being consumed by predators so quickly. And so this is a really important way to improve our detection efforts, is using cages. So we'll deploy a standardized protocol for cages. We're also going to have, again, the protocols that we're making, we are not making in a closed room somewhere. We want this to be a co-designed experience across our community of scientists. So we want opportunities. We're going to facilitate opportunities for feedback. We're drafting protocols now. We'll put them out for a public response period where we can get feedback, integrate it. That'll go through our steering committee and our working groups and the broader Bridge community, so we can really understand what people's needs are, what their concerns are, so we can try to address that as we make these standardized protocols. And then we'll be training on these protocols in our various training modules, which I'll mention in a moment. And of course, the training at CERC in September. Our data working group is also another important piece of what we're doing. This is meant to address bottlenecks for data mobilization, standardization, and sharing. So we're going through that process now. For data and for detection, our goal here, and I just want to highlight this, is not to reinvent the wheel. There's lots of great wheels out there. We're trying to pick one that is good for invasion biology and good for invasion data, and then build capacity around those methods. So we're reviewing data management and sharing approaches now, identifying gaps as they relate to data for invasion biology in particular, and then build on established protocols, help facilitate pipelines for those detection data to go into established data repositories, and to build capacity to again, publicly share those data. So we're creating templates, data templates that can be used with the standardized protocols that we are developing, the fouling panels to start, that people, again, can collect good data, use a standardized protocol, and then know exactly how to get that data into a public repository. And then thinking about how we can continue to build capacity in that direction. our training. All of these things, what we're doing, again, to push out these standardized protocols in detection and data sharing and management, but then make sure that we have a community that can use them, right? So the training courses, as I mentioned, we'll be doing every year. Our first one will be this September at CERC this year, again, focusing on the fouling panels. We're bringing in 20 trainees to start us off, and we'll be doing more trainings on other topics in 27 and 28. Those fellows exchanges that I mentioned before, just again to provide a little bit more information for those early career folks who may be on the call and are interested in engaging. These are 4-week experiences in person, going to a host lab. You can learn a specific method or approach. We are going to fund 2 cohorts of 10 fellows in 27 and 28. So this is primarily for graduate students, postdocs, and other early career researchers who may primarily benefit from this kind of experience. And if there's any people on the call who are really excited to host a trainee, we're very interested to hear from you as well. And of course, we'll be rolling out, again, those virtual trainings this year to facilitate training on standardized protocols, our fouling panels, species identification, some of these molecular tools that we'll be promoting in our protocols as well, and using the data templates that we're developing now. So I'll wrap up. I know we're largely at time, and then we'll go to a Q&A, but I want to give a quick pitch to our upcoming working groups that are going to be launched in future years. So this year we're focusing on detection. Next year we're going to focus on coordinated experiments. And as I said, this is a really, really powerful way to ask large-scale questions about invasion dynamics. And as we know, invasive species, they're not going to adhere to political boundaries. They're going to go where they want to go. So issues in one area quickly become issues of another area. So we need these platforms for coordinated understanding. And these coordinated experiments can be a wonderful platform to do that. So this is an image from a coordinated experiment that we ran across the Americas a few years ago, which we called Panamax, again, looking at the effect of predation really across latitude. And it's this kind of experiment, again, that can be super powerful. I'm happy to talk more about it, but that'll be the topic for next year's working group. Not just predation, but other types of questions as well. And then in year 3, again, thinking through propagule supply and how we can synthesize understanding about vectors to understand more about patterns of propagule supply and therefore invasion risk, because we know there's such a tight relationship between how many propagules are going to come in and the number of non-natives that you're going to see, that dose response curve. So quick pitch for upcoming events. Again, we have those training modules that we're going to be rolling out as early as July. Our call for fellows. Our call for the training for this September has already happened. We've selected our trainees, but we will have future trainings, so keep an eye out for that. But the Fellows Exchange, we have not yet put a call out for that for applications, and we'll be doing that this fall in October and November. So please keep an eye out if you're interested there. And of course, that first exchange is going to happen in 2027. So if you have any questions, you want to get involved, or you just want to stay connected with us, we have a wonderful program coordinator who I think is on the call, Ingrid Watson. You can contact her and our team at bridge@si.edu to just stay connected with us. And I will end it there. Thank you.
Thank you, Amy. Very, very nice presentation, and I think detailed. We have a few questions in the Q&A, but I think we can group the questions because there are a bit too many. We can start one from Petra Rona, who asks whether this research is connected to One Health. For those less familiar with the term One Health, recognize that human health, animal health, ecosystem health, environmental change are interconnected. So, Amy, do you see Bridge as connected to the One Health? Or perhaps more specifically to biosecurity thinking?
Yes, so 100%. This is absolutely framed within the field of biosecurity. One Health in particular is an interesting one. Yes, there's absolutely connections, and I so wish my colleague Katrina Lohan was on the call right now. She's one of the PIs for Bridge. She's also very active with Smithsonian's One Health Initiative. She runs the Coastal Disease Ecology Lab at CERC, and so absolutely there are connections here. Biosecurity is absolutely our framing. We think about how the movement of species, as well as the movement of, as Katrina I'm sure can talk about, disease and parasites and other species that will impact human health, wildlife health, so our natural resources. So absolutely, it is very relevant. So thank you very much for that question.
And highly connected. Okay, we also have one question from Hannah Kazak-Glibov about ballast water, which is one of the most important pathways for marine species introductions. So Hannah asks, how do ballast water treatment works and other environmental risks posed by the treatment itself? Amy, could you please briefly explain this?
Yeah. So the first part, you might have to repeat the second part, but the first part, how do they work? So different treatment systems have different technologies. Two of the most widespread ones are using either chlorination or UV. They're often coupled with filtration. So you might get a filtration and then coupling with the UV or the chlorination. There are other technologies, and of course, all the technologies that are used the ships have to use approved, certified systems that have gone through testing to ensure that they are actually working. That doesn't necessarily mean that they're always working, but they should have an approved management system, ballast water management system. What was it, Pericles? Can you remind me what the second part of that
It was
question
if there are environmental risks posed by the treatment.
By the treatment, yeah. So I will say that they are designed in a way to minimize risk. I'm not going to say that they eliminate risk, right? So the chlorination, of course, if they're using chlorine, they have to go through a dechlorination step, so you're not dumping a lot of chlorinated water into our coastal environments. So that is part of the risk that comes to mind. I think the biggest risk is when they're not working, right? And sometimes challenging conditions mean that they aren't working properly, or maybe they can't, maybe the system is broken, or maybe the conditions don't allow the ships to operate them safely for whatever reason. So there are, it's not a perfect system. It's wonderful that there are ballast water regulations in place. We, again, our thinking and the policies in place for ballast water are way more mature than for hull fouling. And we're getting there on hull fouling. Those conversations are active, right? But ballast water is much more advanced, and that has, I think, probably everyone in the community understands that that has reduced risk, but invasions are still occurring, very much so. So it doesn't, reduced risk doesn't mean no invasions. It just means reduced risk. But those are great questions. Thank you for that.
Yes. Okay, we then have a group of questions around monitoring tools, data, and citizen science. Nidhi Gopal asks about how electronic engineering technologies, software tools, or programming languages are applied in invasion biology and monitoring, or how do research activities include the underwater exploration of coral reefs located on the ocean floor. And Justin Whittle asks about citizen-generated data in Europe, especially whether, okay, maybe we're mixing things a little bit, but especially whether citizen science can help fill data gaps and whether BRIDGE uses rigid or standardized protocols for the citizen science itself.
Yeah, that's both. There's a lot packed into that, so I'll try to touch on both of those themes. One, for detection protocols, there's lots of approaches, as I mentioned, to detection. Of course, fouling panels are not, or just one, and they're one that they're ready to scale, which was why we started there. But to the point of using, I think the person was talking about maybe some image technologies, some other technologies that can be used, ROVs for detection. And as part of the synthesis that we're going to be doing for the working group is to look broadly at the field and try to have as comprehensive of a view as we can across the landscape, and try to highlight the pros and cons of different approaches. Again, so people understand sort of all the tools that are in our toolbox. And to the extent that, one thing that's very much on our radar as we think about emerging technologies and different ways that people do this is the question that we often get is, can we use machine learning or AI? And this is on our minds too. And I think we approach those kinds of opportunities with enthusiasm and caution, right? To make sure that we are using them to in a careful way, in a way that leverages the power of those approaches, but also in a sound way, in a careful way. So I think I was trying to hear the bits of the question, but I think to the extent that there's these other approaches that people are using in coral reefs, deep sea, there's lots of different approaches that are being used, and they're very much on our radar, and we want to include them in the synthesis. The citizen science, I think, was the second one. So yes, so citizen science is of course an important piece of what we do, and so we are in touch with citizen science networks in Europe. We have our own citizen science network in Alaska that our lab runs. This can be a wonderful way to engage everybody, right? Anyone who's an engaged citizen, whether that's a private individual, teachers can engage on behalf of their class, local resource managers can engage. So we actually have one, our program is called Plate Watch in Alaska. So we use these fouling panels across Alaska as part of our citizen science program. And then we train people how to use those panels, and then they can submit data. And we again, with citizen science, you want to make sure that the bar for entry, again, is low, that you're asking reasonable amounts from people. You don't want them to get burned out or overwhelmed. You want them to stay with the program. And as we know, for those of you who are active in this field, species identification can be tricky with a lot of these taxa. So what we do, I mean, of course, in Alaska, it's a high latitude environment. So the species pool, relatively speaking, is going to be lower, right, than some of the other regions, which helps. And we focus on target taxa, which is an approach we're also taking in other regions of the world, where we don't expect you to know everything, but we're going to teach you how to identify some of the usual suspects that we expect to be arriving, either that are there or we expect to be arriving. And if you can reliably identify those taxa, then that also helps fill a really important gap. So I think this idea of having target taxa and lots of people who are trained up to that point that can identify them can really provide a nice layer of information across these large scales. So yes, on the citizen science, absolutely, it's on our radar, and I think we could do more in that space. We're not done, right? I think we can certainly do more in that space to leverage enthusiasm and expertise across the global community.
Yes, one of the major advantages is the social capital and the engagement of the people. So there are too many questions. I will try to follow. There is a golden question here by Precious Titena. What are the most effective biosecurity strategies currently implemented used to prevent marine invasive species? Of course, biosecurity is central, is the most important step that we need to take. But what are the most effective ones?
Yeah. So, and I think you really bring up a good point here, Precious, about prevention. And I just want to, this is the most cost-effective approach to managing invasions is to preventing their introduction in the first place. And so it is very hard once they get into an environment and they become abundant and they spread, it is very hard to put the genie back in the bottle. And so at that point, you're sort of just in a management rather than sort of an eradication stance. So prevention, to your point, is really, really important. So the best policies that we have right now in the US for prevention are those ballast water management convention guidelines, the IMO guidelines, the guidelines that are required in the US that require treatment. Because of the amount of ballast water we get in our country every year, that is important, is critical. We are understanding, again, what compliance is like, how good those systems are working. So again, doesn't remove risk, but it certainly is reducing risk. And that has been fantastic. And we've also noticed in our country how rapid the uptake has been. These requirements have come online in 2017, and we already have about 98% compliance. And at least having a system operating, the reporting, again, whether the systems are perfect, is still, we're trying to understand. But the shipping industry is complying at a really high level. And so when we think about what are the most effective biosecurity strategies, having ones that are required, that have strong uptake across the global fleet have been, I would say that those are likely to have the most effect. I'm really excited about biofouling conversations and trying to think about how we can reduce risk of invasions through management of biofouling. So again, those conversations are an earlier stage, certainly than the ballast water. But I think that will also be an effective biosecurity strategy. There's challenges there too, of course, that we're going to have to work out. But I have strong hopes in that direction. So thank you so much for that question. Yeah.
Indeed, and shipping has done major advancements in the last decades to improve this. Okay, one question, two questions come from Frank Muller Kalger, who asks similar question to what we discussed before. As plants develop to detect alien and invasive species, it will be super useful to capture the characteristic biodiversity of an area. And I know you worked a lot with biodiversity, meaning can we work to not focus on just detecting the invasive species? And another question which I think is related is how does Bridge plan to better link management to policy and science?
Yeah, thank you. Both really great questions. I'll try to touch on them both. So absolutely, this is biodiversity monitoring, 100%. And so it's the focus of Bridge is on non-natives, but it's very much within the context of biodiversity. And when we think about detection efforts, there are ways, as the asker of this question probably realizes, to we can focus on target taxa for non-native species, but we can also get metrics of biodiversity broadly. Doing that, our lab, in our surveys, we get biodiversity data. We look at the whole community, to your point. We understand the value of that, and that's important for a lot of different reasons. It is also a big lift in terms of the expertise you need to have to accurately identify every taxa in a community. We do our best, and we have a large team with decades of experience. So we totally agree with the value of that. The question becomes, how do we scale that? And how do we make it accessible to people, regardless of their area of expertise and resources? One way, of course, that comes to mind is genetic approaches. Can we use eDNA and other sort of metabarcoding approaches to get at that sort of the broader swath of biodiversity without necessarily relying on local expertise morphologically? The answer is yes. Of course, there are great potential as well as limitations with that approach, as with every approach. So these are very active conversations in our Bridge community, particularly when we think about those standardized protocols that we're using. We are going to be rolling out some molecular ones, also with the intent to be able to capture some of that metabarcoding, some of that biodiversity data with it. But we have to have good libraries to tell us what sequences are. We have to have resources to be able to sequence those data. We have to be able to have collaborators that can handle molecular samples, which is not, there are extra steps to that beyond doing the morphological work, as we know. And so, but to your point, absolutely, it is very much within the context of biodiversity, and we definitely want to move in that direction as well, while also building capacity again to understand that non-native layer of invasions. And I know we're coming up on time, but the second part of that question was on management policy, and that is the goal, is to create to accelerate and expand robust science that will directly lead to sound management and policy. And so a lot of the collaborators that we have in our partner networks are managers and policymakers, so they can be part of this conversation really from the beginning. And we're also engaging through our advisory board. We're going to be engaging different leaders within different sectors to make sure that we have that dialogue. So we as a scientific community can be addressing their needs and hearing from them, what they need, and there can be that exchange of information there, because it really, that is critical. And we do that in our lab too. I mean, we work very closely with Coast Guard. We're science providers. We are not compliance entities, but we provide science for our Coast Guard to do just that, to inform their thoughts about management and policy. And those kinds of models, I think, can be used elsewhere where they can within the governmental structure.
And I feel that the globe should take advantage of over the highly invaded regions like the Western USA or the Eastern Mediterranean, because they are natural laboratories in a way, so they can be used to study the marine invasions and give early lessons to other countries. We have a lot of questions that we cannot answer everything in this short time frame, but this shows how interesting the discussion is and how it sparks the interest of everybody. I mean, we don't have that many participants, but everybody asks the questions. Because you mentioned eDNA, I will go to the question of Justin Whittle. Besides eDNA, what other emerging technologies are you most excited for? Um, I guess,
Uh,
guess.
yeah, I am excited about eDNA. And so I think a lot of people in the field are excited about eDNA. I also think that remote image analysis, different using ROVs to collect images, and then using different AI technologies to process those images, I think can be a very interesting approach that we're exploring. So I think that there's some cool approaches on the horizon. And I'm not going to even say that, that I'm an expert in all of them, but we're going to pull together hopefully the right people to have in the room to be able to teach us about different emerging technologies, really as they're just starting to come online, and then be able to see what the opportunities are to roll them out across the community. So thank you for that question.
Okay, I don't know. Do we have to close it at 5? At my time,
I'm
5.
not sure.
Sorry. Okay, we can continue if you are okay and you have some 5
Sure.
or more minutes just to
Yeah.
ask a few questions about participation, about
Yeah.
eligibility and research exchanges. For example, Nidhi asks about opportunities to collaborate in data analysis, whether people can submit a CV and whether there is any remuneration or support, and whether participation is open to engineers and researchers from outside marine scientists. And also, Antonella Rodriguez asks about when research internships or research experience for early career researchers from outside the US might happen. And you can answer this because you already touched on this, but maybe it wasn't clear. I believe it's open to
Yeah.
everybody from around the
Yeah.
world, right?
Yeah, I can speak to that for a moment. And if there's any questions that we're not able to get to today, like any of them, but especially on ones about opportunities, whatever, please do reach out because we're very happy to answer those. So the fellows exchanges, that opportunity, we're going to have 2 cohorts of these exchange opportunities. One will occur in 2027, one will occur in 2028. Each cohort will have 10 early career scientists. The applications for the 2027 program are going to be rolled out in October, November of this year. If you want to be on our mailing list, please bridge@si.edu, shoot us a note, say, hey, I'm interested, and we will put you on a mailing list to get those application materials when they're rolled out. They're open to everybody. We do track, since we are a network of networks, we do track different if you have an associated partner network that you are part of, we do track that information, but we don't exclude people who are not part of a partner network. And we want broad participation. I think one of the questions was, do you have to be in marine science? I think there was a question about, or can you be in other fields? Other fields are great. Yeah, I mean, if this is an opportunity that can help you engage in the field of marine biosecurity and bioinvasions, please do apply. It will also be a process. We'll probably have a lot of deserving applicants. It'll also depend on the hosts that we have, right? And there's a process that we're going to go through to match them. So to make sure that whoever those selectees are, those trainees are, will be matched with an appropriate host. And so that will be part of the selection process as well. But please do reach out if you're interested there, and there'll be an application materials, likely a form that you'll need to fill out. I don't know if there was another part of that question, Berkeley, that I missed.
No,
But
I think it was covered, and they mentioned that it was also already covered by the presentation. The question was asked before, but thank you so much, Amy. One question is Sara Arenas is asking. They're using a similar method and monitoring method with the settlement templates,
yeah,
and they're combining this approach with environmental DNA, eDNA. In Colombia, and they would like to ask whether you complement your approach with a risk assessment, risk identification of the species, like we do horizon scannings, let's say, based on the risk of species. If you complement this work.
yes. Yes, so that is great. So the answer is yes. We do as a lab, so I'm also on the steering committee for one of our partner networks, which is a network across the Americas called Combina. And if this sounds like the person is from Colombia, and so if you would like more information about Combina as a network that engages in biosecurity, please do reach out to us and we can connect you to our program coordinator for that program as well. But we are just now, Combina is also a new network across the Americas, again, focused on marine biosecurity, just started a couple years ago. And we are just, funny that you ask, but we are just now launching an early detection and alert system. We're piloting it in the tropical Eastern Pacific this year and in 2027. Part of that will be early detection, and the alert system will be launched to tell your neighbor when things are on their way. And part of that will be establishing watch lists, which I think gets to your question of like, are you doing a horizon scan? Are you identifying risk? So yes, our intent is to be able to establish watch lists. Those will we have to work through as a community across the Americas, like how we want to approach that. And there's lots of complexities, as you can, I'm sure you appreciate, for having meaningful watch lists and try to understand what are the biggest risks for a distinct region. So I'm not going to, that it's complicated, but important. So we want to tackle that and sort of couple those watch lists again with an early detection system and an alert system to make some headway on this across the Americas. And our hope is that through that process in Combina, that we can create a model for doing this, that we can expand across Bridge, or at least have a replicable model that can be taken to other regions. And so I think this is clearly a space that we can have more kind of international coordination and collaboration on. But thank you so much for that question. That's a great question. Important one.
Thank you, Amy. Great answer. Although I have to say that with the risk assessments, I'm a bit hesitating. I'm hesitant because, you showed in your slides that there is limited data and limited knowledge about the tropical areas, which are the species usually that they are introduced. And by having limited data, we might do risk assessments, and then we might overlook other species, or the species enter new areas and they change. Conditions change, the characteristics are different, so the invasive dynamics are different, and the impacts are different than what we anticipated. And this we have noticed it many times.
Yeah, I agree.
Okay, Suji, last question, I guess, which was quite answered earlier, but it's nice to finish with the collaborative spirit of Bridge.
Yeah.
And Suji does asking if Bridge is collaborating with any overseas organization that have a planning future. I'm not sure what the planning, if it was a typo there, or have a plan in the future. Okay, or do they plan to collaborate with overseas? Yes.
So Bridge is global. And
Yes.
so our reach, so we are a network of networks. And so the networks that we collaborate with, and again, we're continuing to grow, as I mentioned at the top of we are just launching Bridge now, and we're very eager to expand. And so this is not a comprehensive or exclusive list. It's just the groups that we are currently collaborating with. So some of those are US based, some of those are regional, as I mentioned, Combina across the Americas, and some of those are global. We have several global networks, MBON, we have ICES, that's Northern Atlantic. We have the GAME project, doing coordinated experiments across the globe. There's several networks that are part of our Arctic Council. Most of our networks are international in scope, and that's critical to what we're doing. Our intent is to foster those international collaboration and coordination. So 100% yes, that is absolutely the platform that we are trying to build. So thank you very much for that question.
Very much needed, highly important, high priority, and increase in this interoperability across the different nationalities. And okay, they asked for your emails, and Ingrid replied with a bridge at si.edu email. And Frank says, thank you, Amy, Pericles, and the Bridge AR Center and Bond teams for bringing these talks. Looking forward to hear more. Thank you, Amy, for your time. Thank you, everyone, for being here, for listening to this inspiring. And it was inspiring to watch you, Amy, with this passionate talk and passionate character about the topic. And it was exciting. And looking forward to hear more about the Bridge and learn more about what you're doing and catch up in the future.
Yeah,
It was nice to watch you.
Thank you so much, Berkeley, everyone. And for everyone. Thank you so much.
Thank you. Bye-bye.
Bye-bye.
Bye, everyone. Bye.
so much.
All right, good. Yeah. I didn't know if I should just sign off or stay on, but it's nice to see you, Joanna. Thank you again so much for this opportunity. It was really lovely to be able to join you guys today.